Well, thanks for the opportunity to speak today. Unfortunately, I am currently in the Midwest in Iowa, not with you guys today, but I'm excited still to give a presentation on at IDT, Integrated DNA Technologies, the end-to-end solution we've built over the last couple of years to detect CRISPR genome editing events. So this is, really great when you're working on developing research projects or therapeutic projects using CRISPR to go through and truly characterize the level of editing at your on-target site as well as your off-target site, and builds in the bioinformatic capabilities to do this, as a wet bench scientist as well. So just a quick review here. The main enzymes that we work with at IDT are strep pyogenase Cas9 and then AS and LB Cas12a. And these are both RNA-guided restriction endonucleases that make a double-stranded break in the genome. They have unique, distinct mechanisms and structural requirements. Cas9 recognizes an NGG PAM site and makes a double-stranded blunt cut in the genome, and has about a 20 nucleotide protospacer sequence, and is recognized with a cRNA and a tracer RNA that's often fused together into a single guide RNA. Whereas Cas12a recognizes a TTTV PAM and makes a staggered cut, and this is a much shorter complete guide RNA oligo or molecule ranging in the 21 to 24 nucleotide for the specific protospacer sequence, and this ends up being about a 40 nucleotide complete RNA molecule. So these are the two main systems that we study and are most widely used in the field. And so the analysis tool I'm going to discuss today supports both of applications with both Cas9 or Cas12a. So once you introduce the CRISPR reagents into your cell and a double-stranded break is made, there's two main dominant repair pathways that the cell's endogenous DNA repair systems will undergo. If you provide additional donor template to make a correction, the homology-directed repair pathway can take effect. Otherwise, often if you're just introducing a double-stranded break, the non-homologous end joining pathway is the predominant. This is error-prone and can then often insert insertions and deletions, which leads to a frame shift knockout. And so this is the pathway we'll mostly be focusing on today, but this is, this is error prone, and there is chance for off-target effects that we want to make sure we're looking into today. So that'll be a lot of the focus. And so CRISPR genome editing, like many other oligotherapeutics, is imperfect, and there can be off-target effects that occur. And so here's a typical output that's been published pretty widely. The GuideSeq technology is one example of an empirical off-target nomination tool that, along with the CRISPR components, introduces a double-stranded tag that could be inserted at the site of the break, and that you can then, through adapter sequencing, go and detect where this is integrated. And here's a common output that shows the on-target site, the level of NGS reads you're getting, and all other potential off-target sites where we're detecting that tag. So this is a common method and technology used to determine where there's potential risk of off-target editing associated with your CRISPR cargo. So it's very important to select optimal guide RNAs and to be able to assess what the effects of these guide RNAs are, both from the efficacy of how efficient this guide RNA is, as well as if any of this off-target editing is happening. And so there are many in silico prediction tools out there that help you design your guide RNAs and can do some predictions for if there is a risk of off-target editing. So that's typically, where you start in your CRISPR experiments, and then you need to go and you need to do some empirical on-target performance and verify that this is an efficient guide RNA. And so, there are many different methodologies for doing this. A lot of, fast methods such as PCR based or endonuclease or next generation sequencing. So once you have your highly active guides, it's then important to and you've kind of settled on your guide RNA target. It's important then to appreciate what the off-target potential is. And so that's where like the technology like GuideSeq, there's other technologies such as ChangeSeq, CircleSeq, there's your in silico predictions, but it then is important to go and empirically look and see what the potential this particular guide RNA has for, editing in other particular spots in the genome. And so once you do that, you've got to then have a way to measure this also in your cells of interest. And so I'll talk about this later slides, but the RAMP-seq system is a technology that IDT has developed. In RHPCR, we developed back in 2013, and I'll go into more detail on that. But this allows you to then look at the on-target site that your guide RNA is cleaving at, as well as any potential off-target sites in one high throughput NGS reaction, and do that on your cells that you've actually edited. So we'll go into a lot more detail on that, but this is kind of a high level overview of how we select guide RNAs and the steps to go through for assessing their function. So here is the kind of shows a case study of just how important it is to quantify those off-target effects, and by which reagents and tools you use can affect that too. So here's a safe harbor site AAVS1 and a guide RNA in that region where we have here delivered the guide RNA into cells that stably express Cas9. So there's continuous expression of the Cas9 complex, which often results in larger amounts of off-target editing. So here is looking at the all the detectable off-target sites that come from this type of system. And so you can see that there can be high levels of unintended editing. The on-target, you're seeing 80% editing, but at all of these other 28 other sites, we're detecting editing at various levels. Switching now from delivering into Cas9 cells and delivering your Cas9 as a recombinant protein, so delivering a ribonuclear protein complex, really reduces the amount of sites where we're seeing that off-target editing, but there is still some present. And then, as we published in 2018, IDT evolved a high fidelity Cas9 that can maintain that high on-target activity that we saw at the on-target site for this particular guide RNA, but we've now significantly reduced the off-target editing. And so the nomination of which sites to look at through your various methodologies, and then going and actually quantifying those with an amplicon-based sequencing technology, for example, is really important. So this highlights here, the different strategies to do editing, and then, going back and verifying the level of risk that you have from that guide RNA site. So now I want to shift a little bit and talk about actually quantifying the editing at the on and off target site. So there are a number of methods that exist, like I mentioned, that can determine the editing, and some of these are fast and cheaper methods, like shown on the left, and such as T7 endonuclease assays or heteroduplex cleavage assays. And these work well for high throughput, but their accuracy and sensitivity is very limiting, and there's not an ability to multiplex and look at those on and off target sites at the same time, and obviously not providing allelic information. And so here's something from the literature just showing that you're really misrepresenting editing in some cases, and this can be due to the nature of the editing population, T7's inability to recognize one base indels, and et cetera. Additionally, in the second panel, this is comparing data adapted from the Shapiro et al. Publication in 2020, where we're looking at Sanger editing, looking at just on-target editing from using either ICE or a TIDES analysis system to NGS. And while Sanger editing or assessing your editing by a Sanger methodology is also high throughput and can get you more information, it also doesn't allow for the multiplex capabilities, and it's not as sensitive. And so what I mean by that is, you see a good correlation until you really get down to these low percent editing levels, and then the variability really starts to show. And so you just can't get as sensitive, which is really important when you're looking at these potential off-target edits that may be, in the under 1% range. So the last panel on the right, which is in a manuscript we recently published at IDT, shows where we took spike-in controls where we knew exactly what the various levels of editing were, and then had our analysis tool predict this following NGS sequencing. And what this shows here is that we're able to get down to pretty low levels of detection of indels at these various control sites, down to levels of 0.1%, which is around the background noise with Illumina sequencers. So what exactly is the RAMP-seq CRISPR analysis system? This is something that we've evolved over many years. And so I'll go into the detail in the next slides, but this has really now become a complete, end-to-end process for doing your editing analysis for CRISPR. So we've now got the design of your primers through a bioinformatics tool that we've developed. We've got the library prep technology to do the next generation sequencing that can feed into an Illumina platform. And then now we've got an analysis tool that comes with a user-friendly GUI and a really high quality graphics that can provide a lot of various interpretations that we'll go over here. So first, let's start and just talk about what the RAMP-seq chemistry is and how to design the primers to look at your editing associated with the guide RNAs you're using in your CRISPR experiments. So I've mentioned this a few times, but what is RAMP-seq? And so RAMP-seq is a highly specific multiplexed next generation amplicon sequencing technology that uses RHPCR. And so RHPCR is an amplification method that uses primers that contain an RNA base and a 3' blocking group, shown here with the green circle. And so RNase H2 is present in the PCR master mix and recognizes this RNA DNA heteroduplex and cleaves, releasing this blocking group and allowing amplification to occur. And there's a second round of amplification that adds on the P7 and P5 sequence adapters to create next generation sequence ready amplicons. And what this really does is it allows you to have a primer pool of up to 5,000 different assays in one PCR reaction. So it really reduces your time of setting up all of that PCR and doing one library prep. And then this allows you to, for CRISPR editing, taking your edited genomic DNA, look at that on-target site, as well as look at any predicted off-target sites that have come from in silico predictions or any sort of empirical nominations that you've done, and do this all on your actual edited DNA that's for your test system. So the work done here has been to develop the RNase H2 master mix and PCR technology, as well as our bioinformatics scientists in the R&D group at IDT have developed this design tool to do all the work to design these, up to 5,000 assays that can work well together. And so there's a design tool available that you submit your guide locations using either bed or FASTA files. And in the bottom left is an example of a bed file that contains information like chromosomes, start, stop, strand information. And you input this into the design tool. There's an option to select CRISPR editing analysis, which will then make sure that the amplicons are designed using characteristics capable of determining CRISPR indel editing events. And then there's additional parameters in here that you can select your reference genome. If you have size limitations that you'd want to infer here, as well as to pool your designs or not. And if you're looking at this for a multiplex application, you know, you would ideally want to pool all of those together. But if you're using this to just look at a single on-target site, which I'll go into more detail on, like if you're doing a CRISPR library screen and you're looking at thousands of individual guides, not pooling these will allow you to essentially get, you know, one PCR primer designed for one respective guide RNA. So it supports, you know, both of those use cases. So kind of what's going on under the hood for this design tool is when you feed it all of the different targets you want assays designed for, is it's going to then do several rounds of exhaustive primer design searching and in silico QC to determine and select which primers, which assay pairs will be able to be pooled together and or need to be separate in various sequential steps. So for applications like, you know, looking at on and off target editing, it's desired to have, you know, a pool with as many of those primers in a single pool as possible to really simplify the amount of PCR amplifications you're going to have to do. So what it does here is it goes through and it selects the best primers for every target site. If there's a primer that it just can't react well with the other primers, that this one will be set aside and it will be tried to put together in a secondary pool, or this will be indicated that this is just a single reaction that will need to be run on its own. And so that's all kind of part of the report out that the analysis tool will give you when it's doing the design. And additionally, like I mentioned, there's also the ability to do the non-pooling, where if you're looking at, you know, CRISPR gene knockout screens or doing some sort of like large DOE type experiment where you're looking at various different HDR controls, you can then have a single primer pair for every, you know, associated treatment of your, you know, library or your respective HDR experiment that you may be doing. So then next, focusing on the actual amplification, the library prep. So we've got the primer designs then. First, I'm going to show an application of using this for that singleplex application if you're, you know, looking at editing from a guide RNA library. So this may be where you're at the first stage and working to pick which is going to be my lead guide RNA candidate to further study for my therapeutic target or for my disease stage. And so the RAMP-seq design tool then allows you to, you know, input in the various guides that you want assays designed for. And so you can think about this, you know, on a 96 well plate. It'll give you optimal assays for each guide RNA location. It'll consolidate any if they're within the same, you know, within a certain window where they could be amplified with the same assay. And then essentially, you know, give you a report to get a plate of guides that, or a plate of primers that would match your, you know, your plate of guides. And so we've compared this to other widely available technologies. For example, here shown Primer 3 is a common design tool used, and other available master mixes, just to highlight how the RAMP-seq master mix and PCR amplification works comparatively, especially in just using this as a singleplex. And so the user interface is really slick and allows you obviously to multiplex, and then it adds on this, you know, ability to have that CRISPR specific design. So it'll, you know, take into account the indel events that could occur and get the correct window size there, and allows for that batch design submission to, you know, account for, you know, if you're looking at thousands of guides at once. And then additionally, it allows that multiplex capability. And so once you've got the primers designed, what we're wanting to show here is just the robustness of using the designs that come out of the RAMP-seq design tool. And again, this is just focusing on a single amplicon here for looking at, you know, what would be a library of guides, and then, you know, sequencing every well here. And so in this panel B, what we're showing here is the RAMP-seq designed primers is indicative of the plus in the first bar, and then with the RAMP-seq master mix. So, you know, we're getting over 90% amplification success when we've fed this 96 targets in this case. the teal color, I guess more aqua blue in the second bar is actually taking designs from the Primer 3 design tool, and then also using the RampSeek master mix. So this shows that this is the master mix, you know, if you have designs from a different platform, is also compatible with other designs that it would only, in this case, be, you know, DNA only. And then lastly, looking at Primer 3 design, in this case with a Q5 master mix. So showing the highest amplification efficiency with using designs that came out of the RampSeek design tool with the master mix. And just to kind of highlight what the different primer structures are that we're looking at here is, since these are just single amplifications, that, you know, that RNA base and that blocking group really is there for the multiplex capability, but works just as well in singleplex as, you know, a DNA only primer here, but just kind of highlighting what the differences are of those different types of structures of primers and how that activity compares. And then lastly, now actually taking these and doing the sequencing and looking at the actual CRISPR editing between the two different examples shown here, Primer 3 designed assays or RampSeek designed, it's very highly concordant. So very amenable as a, you know, in this case, a very like plug and play method for generating NGS libraries, and then allowing, and this then funnels into the analysis tool, which I'll go into more detail in the next couple slides. The last thing to kind of highlight about the RampSeek technology, which going back to the ability to multiplex, is really just here showing some data regenerated in our lab about the really strong ability to highly multiplex many assays together and not get the primer dimer amplicons that would be associated with if you were using DNA only primers. And so this gel image on the left panel here is a 990 assay panel where we've amplified 3 different genomic DNA samples with just primers that are DNA only. And you can see around the, you know, 200 base pair mark, you know, the dimers that are occurring and that eat up, you know, your sequencing reads. Whereas with using the RampSeek technology, we don't see any of that primer dimer formation due to the, you know, specificity of having that RNA, that blocking group and the RNA's capabilities. And then the panel on the right is showing the high uniformity that can be seen here. And so these are, you know, the read depth coverage for this 990 plex, showing that greater than 91% of the sites amplified have coverage greater than 0.2x of the mean coverage. So I've focused on the design part and the actual amplification. And so, you know, the latest technology that we developed and then tied to RampSeek is now having an analysis tool to do the actual analysis of these, you know, sequencing runs. And so this was specifically designed to have a user interface and not need to run locally on computers, and to be really amenable for if there's not bioinformatics resource or resources available, or the need to run command lines. And all of the background and information about the analysis tool is recently published. I've highlighted that publication here to read more about the development of that. But, you know, why did we go and create a CRISPR NGS analysis software? There's a lot that exists. And the short answer is that, you know, talking with a lot of scientists and just our own internal R&D teams, a lot of these other tools we found difficult to use if we didn't have strong bioinformatics resources or backgrounds, and that they weren't necessarily fully characterized or validated for high throughput workflows. And so what we've developed, you know, really allows for you to do your CRISPR editing and then get a really clear representation of different repair characterizations that have occurred, if there's indels, deletions, repair profiles, compatible with both Cas12a and Cas9, what HDR events have occurred. We've done a lot of work to optimize the accuracy of the calling, as I've highlighted already. And then this kind of, and then this all feeds into then an output that is really intuitive to know what's going on, to take your FASTA files from your sequencing and drag and drop that into this tool and be able to put some sample sheet information and get a clear idea of what's going on at both that on target site, as well as if you're looking at those, hundreds to thousands of off target sites. And the panel on the bottom left here shows that in green this is where we've taken 600 synthetically generated data set sites where we know what the total editing is and compared what was actually coming out of these various tools and just the high level of editing accuracy that we're getting now with the improvements that we've made to this bioinformatics tool. And so, just briefly to highlight, we IDT partnered with Illumina to make this available, not just to our R&D teams, but now externally. And so this is available as a cloud-based web interface that comes with all the necessary, security and compliance pieces, allows for, like I said, batch submission, high graphical interfaces that come out of this. And it's pretty straightforward, where you can drag and drop your FASTA files from your sequencing, add some information in the form of a bed file if these are multiplex, or information about your guide and amplicon sequence if this is individual assays you're looking at, and then be able to get pretty quickly get some graphical outputs of what's the percent edited, what the repair profile looks like. If this has created a frame shift, it's compatible with IGV plots to look at allelic information, and then also exportable files that can give you a lot more detailed information to create various reports. Another nice feature that we put into this kind of goes back to, again, starting with where you're developing, which is your lead guide RNA candidate that you might move forward to studying. And so looking at, hundreds of different guide RNAs at once, we've now got this, it's compatible with a 96 or 3D4 well output where you can essentially see every well. And I've just shown 15 wells here, but it will give you this representation. The size of the circle is determining how many reads that were associated with that particular NGS sample. It highlights if the samples are edited, what percentage are edited, if there's a frame shift that's occurred, if you have an HDR template also in there, what level of perfect HDR. So it's just a really quick way to look at that, and then you can look into more detail actual allelic information as well. So that's really the system that we've developed, and I wanted to take in the last couple minutes and just show like some of the internal data that we've generated to characterize the sensitivity of this system. And so often we get questions about how many reads we need to put at each sample and how that affects the accuracy of the editing. And so for starters, we wanted to look at the number of reads that should be obtained to quantify editing, with confidence at your target site. So here we looked at a panel of 91 targets that were edited under variable experimental conditions and quantified editing with the reads generated with our CRISPR analysis tool, and subsampled the reads of each target in triplicate, and then calculated the deviation of indels quantified with the subsampled reads against when all reads were used. And so on the left, we can see the magnitude of indel fold changes really decreases as you obviously put greater read depth on. So when we're getting to read depths approaching 1,000, paired end reads, and in this case, it's from a 2x150 Illumina MiSeq run. We're really looking now at, it appears that approximately 0.5% of the indels can be annotated with a deviation around plus or minus 0.2% when we're in that, around the 1,000 paired end reads. So we, routinely use at least 1,000 reads per site for indel quantification of 0.5% editing levels. And so next, we wanted to quantify the level of background indel rates that we observe in unedited samples. to get an idea of what the background noise looks like when you're using the RAMP-seq CRISPR system. So we did this with 270 unique loci and 2 cell lines where we gave greater than 10,000 reads for these, unedited samples. And here on the x-axis, you can see the percent indels in the control samples, and they're binned in 0.1% intervals with the relative frequency of all the control samples plotted on the y-axis. And so here we have essentially identified that approximately 98% of the genomic sites have an indel signal below 0.4%, and have thus proceeded to use this to create an assumption that most samples should have indel noise levels between 0 and 0.4%, which comes into effect when we think about our sensitivity of the system. So finally, to see how sensitive the RAMP-seq CRISPR analysis system may be for quantifying those low levels of off-target editing, we created this truth set essentially with variable levels of indels spiked in to 20 unedited genomic DNA with backgrounds that ranged from that 0 to 0.4%. And so we had edited genomic DNA spike-ins ranging from 0.05% up to 3.5 indels into that unedited background. And so to provide a binary call if a sample was edited or not, we performed a Fisher's exact test on the paired end, comparing the read count of wild type and then indel containing reads for the either treatment versus control. And this then allowed us to get a p-value, which we used as a threshold to qualify anything with less than 0.05 as a truly edited site. And so essentially what you can see here is that with 47 to 56 pairs per indel dilution, we're able to correctly characterize 95% of true positives down to that 0.5% indel data set. And that's taken into account that the indel background noise was less than 0.4% and that the reads were greater than 5%. So that's a key requirement to be able to make that claim here that we've shown. So to kind of wrap this up, the RAMP-seq CRISPR system can really now take any nominated off-target sites that you've predicted or empirically defined and quantify the level of editing in your sample, as well as look at that on-target editing. Do this in a in a multiplexed fashion, as well as looking at single amplicons if you're at the stage of identifying your guide RNA and doing large screens, as well as now have an NGS analysis solution that ideally should be a straightforward workflow without needing to have any code or bioinformatics additional resources tied to that. And again, like I said, I'm in the R&D team at IDT, which is in the Midwest, but Sasha Thomas is the local representative for IDT in the UK. So with that, thank you, and I'm happy to take any specific questions or happy to be reached out to via email.